Prokaryotic DNA contains clusters of sequences that work together to regulate and express themselves according to changes in the organism's environment. These clusters, taken together, are called operons. The lactose operon, hereby referred to as the lac operon, is one such cluster found in E. coli bacteria. The operon contains three structural genes, LAC-Z, LAC-Y, and LAC-A. LAC-A codes for transacetylase, an enzyme thought to help remove waste from the cell. LAC-Y codes for permease, an enzyme that facilitates the permeation of lactose into the cell. And LAC-Z codes for beta-galactosidase, the crown jewel of the operon, which converts lactose, a disaccharide, into its constituent monomers, glucose and galactose. The structural genes are vital for the use of lactose as the primary energy source by the cell. However, lactose isn't always the preferred source of energy, as the cell could very well be in the presence of glucose and could therefore be wasting energy by producing enzymes it doesn't need. How then does the cell turn the operon on and off? The regulatory elements are upstream of the structural genes and consist of a promoter element as well as a sequence called an operator. Additionally, further upstream and totally separate from the operon sits the LAC-I gene, which codes for a DNA-binding repressor protein. The LAC-I repressor binds to the operator of the regulatory region of the lac operon, preventing transcription from taking place. However, when lactose or a chemical mimic is present in the cell, it binds to a site on the LAC-I repressor to induce a conformational change, releasing the repressor from the operator and allowing transcription to occur. That way, when there is no lactose in the cell, enzymes required for lactose hydrolysis are not manufactured, but when there is lactose in the cell, the enzymes are manufactured. What if the cell exists in plentiful glucose as well as plentiful lactose? Glucose is the preferred energy source of the cell, so how does it turn off the lack of transcription in this case? Well, an enzyme called adenyl cyclase produces a molecule called cyclic adenosine monophosphate, or CAMP. Adenyl cyclase function is negatively correlated with glucose levels. As glucose levels rise, adenyl cyclase activity is inhibited. CAMP can join with yet another molecule, catabolite activating protein, also known as CAP. Together, they form CAP-CAMP. The lac operon, along with the promoter and operator, also contains a CAP binding site. Without CAP, RNA polymerase can't efficiently start its work. Furthermore, without CAMP, CAP can't efficiently bind to the CAP binding site. Therefore, when glucose levels are low, adenyl cyclase activity is high and produces ample CAMP. Plentiful CAMP can bind to CAP and allow attachment to the CAP binding site. In summary, the lac operon is regulated both positively and negatively. Transcription cannot occur if there is no CAP bound and is further inhibited by the presence of the LAC-I repressor. However, if there is CAP bound and there is ample lactose to release the repressor, transcription will occur.